All right. Hello, everybody. Welcome to the final day of the Applied Bioinformatics in Agriculture and Medicine seminar. And today we are in the final day of the workshop, and we hope that you will. We hope that you have learned something from yesterday and the day before yesterday's workshop. And today we will be adding a few more details and also realizing what we do once you have your BAM files, what to do next, and how to call variants, and then move towards how to construct phylogeny. So please have patience. If you have any questions, feel free to put it on the chat box. We will try to reply that as soon as possible. With that being said, I'd like to ask Dr. Ananta Acharya to lead today's session.
Thanks, Dr. Paudel. Today, we'll start from the files we created yesterday. And before, today is the last day. Again, as always, we are really excited. And also, we are kind of, okay, let's see towards the end and let's see what's your feedback, what will be your feedback, and how can we improve this one, or what to do next, and those kind of things. So sometime in the after maybe half an hour or 1 hour, you will get some email from us for a survey. We ask you to provide the feedback on the survey. That will really help us on how we did, and then knowing what to do next. The other question I'm frequently getting is, okay, sometimes we talked about rice, sometimes we talked about coronavirus, this is agriculture thing and those kind of things, and what about animal genomics, what about NGS and those kind of things. So all the things we are doing right here are NGS. So basically this sequencing, these most of the thing are NGS. So you can use some of the old first generation sequencing stuff from the Sanger. We might use that, but we haven't used that. In the future, we can also do let's say sorry, I forgot about I was trying to say something, but and also in the animal side, the reason we are using the coronavirus as an example is because it's a 30,000 base pair genome. And look, when we are running some data, we are running that. The mapping was finished in few minutes or few seconds. We could demo the BAM files and everything. That is the reason we started with the coronavirus, and coronavirus is the hot topic. But we can do all these things in any genome of your interest, if we have the data. And the other problem is the data. For coronavirus, we have so much data right now. And for a lot of the bacterial species, we will have the data. For rice, we have the data. For corn, we have data. Major crop species, we have data. If you look at the animal side, I think for cow and pig, we have pretty good data. And then if you go beyond those, then it starts, do we have enough data on those species? And there is one fundamental difference in animal genomics, in the genomic selection and in the plants, is seems like in animal genomics, they share their data, and especially they generally, rather than using the NGS or whole sequencing, they generally have that 50,000 or 100,000 marker set. That can still call the SNPs. They will just sequence particular SNPs. They have made some model for genomic selection, and then based on that model, they create so much of the SNP data. And that's how in the animal genomics, but mostly in the pig and cow, that's how it works. So again, whatever we are doing here, we can apply this for any of the crop, any of the live, any of living things, even fossils, right? You have probably heard about, okay, we got the sequencer of maybe dinosaur. So that is the exact what we can do. Okay. Now let me get started with where we were yesterday. And again, feel free to ask any questions. All of us will help you answer some of the questions. I saw one question a little bit earlier about how to download the FASTA file or the reference file directly. There is some utility called, I think, Intraze. I showed the SRA. That is the reason is because we would have so many SRA. That is why I saw the showed FASTQ DOM. Generally for the reference genome, because this is just one reference genome, we generally don't download a lot. So I saw we just we can just use the wget and we could do that. So that is the reason we stored. But you can do some other techniques and download it directly. Okay.
Yeah, adding to that, if you are interested in plant genomes, there is a site called Python. So you can go to this, and there's a lot of different plants whose reference genome is kept there. So if you are interested to download just any of these links, and you'll be able to download of the genomic data from here. It's called Phytosome.
Does it allow downloading with wget or directly on your terminal?
It will also allow.
Okay. Okay.
Yeah, it will. You'll need to log in. So it's just the thing, an account is free on this, so we can just do. It's like fine. So here you can see the annotation file. These are all the annotation file that is for pineapple. It shows the assembly. This is where the reference genome would be. So .fa.gz, this would be the file that you will download if you want to download a RIP genome. So if you just right click and copy link as here, you should be able to go to your mobile external and directly download it.
Okay, that's great. And you using the wget, right?
Yeah.
Mm-hmm.
I hope it will work. Yeah, so we can directly download. Oh, maybe it didn't download. Maybe
Yeah,
yeah.
I don't think so. Yeah. You might have to click it again there.
But yeah, if you click it, you can just directly download to your own computer and then transfer it.
Yeah. So that's why there are different methods. One is you can download your local computer and then transfer from your local computer, or you can use that Intrazio utility over there. Okay. Now let me share my screen and go from here. And while I'm demoing, if you see any questions, please feel free to stop me and reply. So let's remember what we did yesterday. I think the last one we had yesterday was we mapped some of our file, one of the pastel file, one of the sequences to the reference genome, and we got the SAM file, and then we also converted to the BAM file, which is the file. So if we now let's do that again. That's how you see it, and this is exactly what I showed yesterday. So this is that, this is the sequence name, and you can see this is ordered in the genome and those kind of things. So what we are going to do today is, and also you saw yesterday that when we are playing around, we saw some of the sequences had SNP, and in one position, almost all samples, all different reads had that SNP. So what we're going to try today is to call the variant on our file. So let's say we are going to call the variants on this file. So the command for calling the variance is we'll use the mpylab. So this is one thing that somebody was asking yesterday, or maybe if we have some history of your work, you can actually use the arrow key and go back to what you have done earlier, or you can actually just type history and see what you have done. So what I want to show here is try to see, rather than probably I have already done today or yesterday. So I want to see what was exact command. And remember, in the commands we have so many different options that sometimes we have to make sure that we get the right format and right options. So if we do here, let me see the history, and let me find something I did the mpylab. So if you look at this, these are the different commands looks like I did yesterday. So I can do the exact same thing today, right? So so the command here is I was muted. This is the reference, this is the reference, our BAM file. We want to call the variance, and if you can actually output this mpylab format in the mpylab format or the VCF format. But today I'm going to skip the mpylab format because most of the time we use the VCF format. So I'll directly call the VCF from this one. So how I would call the VCF is, again, whatever is that output, I'll command that, pipe that to VCF tools call, and this is something that I may not have time to explain all different options here, but what we can do is these different hyphen and options you see, these are actually different parameters. Okay, what is the format you want, or what are the different kind of variants you want to call? So whether it should be, you have to have all 29,000 bases, you want the sequence on that, or you just want something that is different, and those kind of things. Okay. So when we send out the course today or tomorrow, we'll make sure we also link to this program so that you can look at the manual. And this one is SRR 1792, so I'll use this one as our out file. So now it is calling the variance on this one. And look, some of the messages here, it is saying none of the samples file or ploidy file is given, so it is assuming it deployed. So why and we know it is coronavirus, it's not deployed. It's just, even RNA is a single stranded RNA. So but it is assuming deployed. So why does it matter? Because this is the statistics. Most of the variant calling softwares use some statistics and call the variant. So for example, we have 2 copies of for every chromosome we have 2 copies, right? So when we have 2 copies, let's say when we sequence, let's say we saw something at one of the allele is 10 times, other allele is 1 times. Is that a true variant or not? It's kind of confusing, right? So 10 and 1, it looks like it is variant. If it is a pooled population, however, 10 and 1 in a deployed population, maybe not. If it is a random, or 50 to 1, might be random. Or what about just 1 and 1, or 2 and 2? Then that might be real. So it uses the depth of sequencing for everywhere and calculates statistically whether if it sees 1 miscall, 1 kind of snip, it might be sequencing error or other things. So it tries to call it based on that statistical method. We generally use 2 programs. One is this VCF tools, or also SAM tools. These are from the same programmer. Previously we used to use SAM tools. Right now we used to call mostly BCF tools. Same similar algorithm. The other very popular one for calling the SNPs is GATK. So these are the two softwares we generally use. Okay, so it is taking time. I don't know why it's taking that much time. Maybe a lot of you are working on. So what I'll do now is I'll stop this one so that I already have that file, so I can show you what it would look like. Okay, so if you look at this srr2.variance.txt, I don't. If you look at the size, it doesn't have anything, right? So that's why we know this is not the. We don't have all the results. So not to confuse, I'll just delete this one. And if you remember Mr. Subedi's talk, you can use the remove command to remove that file. Now I know the variance.bcf file was for that file, and I created that earlier. So I'm just going to look at that file now. So the way I look at that file is, again, the file format can be VCF, SAM, FASTA, FASTQ. Basically, they are all we call flat file. The flat file are basically it's the text file. So it is still the text file, but it just has a different headers, different columns, and that's how it makes a different file. So by mistake, instead of writing the variance.vcf, if you write variance.txt, that is still going to work, but it will be you will be confused on what that file is. So now if you look at the VCF format, and again, remember Dr. Paudel's talk 2 days ago about the different file formats. The file format here is VCF 4.2. It has this different ID or this indel, IDB, DP, these different things he talked about that day, right? And then you have this different info, and if you remove all this that starts with hashtag, now we used to call SARP or hash, now everybody calls hashtag, right? If you look at that one, and so this is the end of the file. So something very interestingly, if I start just looking at this line, there is just one line that is different. And if you look at this one, at the position 24034, it is supposed to be C, but it is T, and this is the quality, and this is the depth of the whole that line. So it is a 250 base pair 200. So this was sequenced at that location for 250 times. So if you looked at the VCF file yesterday, I mean the BAM file yesterday, it would probably sort 250 lines at that location. And if you look at that 250 lines, and now you can forget about all this format. It has all these different, everything has different meanings. So you can go ahead and look what the meaning of that. But what we really care about is here it is saying GT and PL. So this is the genotype, and this is the probability score. So if you look at the genotype, it is 0, 1. That means it is heterozygous for that location. So it has a C and T. Both you can find both C and T on that location. Okay, so I'm going to show that again for some other VCF file. So I also had VCF file that is probably it was India or Bangladesh, one of those ones. So if I look at those one now, if you see this one, now it is the end of the file. So there is just one line. If you look at very closely, it has now more than one line. It actually has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 lines. So there are actually 12 mutations in this file. So you can always go back. Somebody can always go back and source what this one is. Maybe go in the NCBI and start finding out what this was, whether it is from Bangladesh or India or even US, and when it was sequenced.
Dr. Acharya?
Yep.
Yeah, I just want to point out that this 23403, if you look at it, this is the D to G mutation that we discussed earlier, A to G.
Yep.
Yeah, so this is the one where infectivity is really high, and people are studying right now to see what the pathogenicity could be. So I just wanted to point out that. Thanks.
Yeah, great point, and thanks for reminding. So again, what Mr. Subedi is saying is, it was in the reference genome, it was A, and then it is converted to G. But we also talked about it was a D to G mutation, but we are actually seeing here A to G, right? So the difference is, when we are talking about the D to G mutation, that's the amino acid, that's the codon. So what happened is, this A changed to G, and whatever used to, let's say the ACT probably coded for D, but GCT probably coded for G. And that is the reason we had that D to G mutation. And also in that look at that one, we say 614. That was the 614th position of the S gene. However, in the whole genome level, this is the 23403 position on the whole genome. And that is the reason why that there is 614, why there is D and G versus why it has A to G in this position. Great point. Thanks, Mr. Subedi. Okay. So what we are seeing here is from the very beginning, and as I pointed out, the sequence from Nepal that was done in I think January or February, and that was the direct one of the patient who came from Wuhan to Nepal. At that time, there were no mutations, or just that one mutation. But right now, it has been mutating so much that we are seeing this much mutation in recent samples. So can somebody please help me find out what this sequence is? SRR 1180 1823. If we source this in the SRA NCBI, we should find out. Okay, so now let's move on from this one. So now we know how to call the variants from this one. And yesterday we had some questions about what do we use a different alignment? What is it? Why do we align with the BWA, then calling the SAM tools, PCR versus our pairwise alignment or multiple alignment. So now we'll move to the multiple alignment. Okay. So let's look one of the file Mr. Subedi showed 2 days ago. Now, if you look at this COVID samples on .fasta file, you see there is one reference one, let's say 1, and then you will see the sequence. We know this is the FASTA sequence, not because it has the extension FASTA. We actually know it is a FASTA because of the format. So that's what I'm talking about, is probably what you write as extension probably doesn't matter. It's for your reference, but what is inside should be a good one. So think about this one. You made write a Word file, and then you save you change its name to something dot instead of DOC or DOCX, you are saying XLS or XLSX. Then it is not going XLS. It is not the Excel file, right? It's not going to be opened by the Excel file. So because it has a totally different algorithm at looking at that. Okay. Also, Mr. Subedi showed us how to find out what are the sequence names on this one. And if you remember that, grep, that's this comment. This is just the regular expression. Okay. So I think the full form of grep is GNU regular expression pattern, something like that. when we use this caret symbol, that means always look for something in the very beginning of the line. And then what we are looking in the very beginning of the line is always that greater than sign. And we are looking in this covid.samples.fasta. Okay, so if we look at this one, so what we are going to do here, what we are doing here is we actually have a reference genome, and then we have other sequences, one from Nepal. We also have 3 from India, Pakistan, Italy, USA, Sweden, England, Australia, different stuffs. And thanks to Mr. Subedi, he wrote a script that you can give a file name, and then you can say, okay, how many coronavirus examples you want from a different country, and he has a script that you can actually get that from a GIS. Yes, and that is where that bioinformaticians come. We don't only just do the scripts that is already somebody wrote and just run that. We also have to work on our own script. So that is something that I think this is the select by countries.py. So this is the Python file, and I will just briefly show how it will look like. We are not going to talk about the Python here, but just briefly show what we generally write. We have to. So if you are a bioinformatician, you might not only just be doing the things I was showing earlier. You might also be doing this one, and this is really important because you don't want to go in the NCBI and click and download everything manually. So as a bioinformatician, you always want to automate the things. So this is that script. So it looks like a simple script. What is important is you get the logic right. Again, when in the programming, I always say, know what you are doing and get that algorithm right. Programming language doesn't matter. If you know one language, you can easily learn the other language. And it takes this input.csv file, which has, okay, Nepal 1, India 3, Pakistan 1, and that's exactly why it has the exact those number of sequences. Okay, so you already saw how the mapping works, right? How we do the mapping with the BWMM. So what I'm going to show now is you can also map the FASTA files. You don't have to just map the FASTQ files. So I already did that, because this is a huge, big file. So I also mapped this COVID samples file. Okay. So when I mapped those files and made a SAM file, this is how it is going to look like. There is a reference. This is what our sequence was. This is the again reference genome, and this is all this sequence. And this is a really long sequence, right? About 29,000. So again, something that's how a lot of the bioinformatics do. We don't rely on a single tool. If you look at here, you will see minimap2 and this different command. In the previous one, you probably saw BWMM and the command. Now I use the minimap2 because it is a longer sequence. I think minimap2 is better for longer sequence. So again, we have so many open source programs. You can use a lot of them. So for NGS though, for NGS where we are talking about the short reads, less than 500 or so base pairs, we almost always use BWA or BOTI. And minimap2 is now really popular for using with the Oxford Nanopore or PacBio or whole genome to genome comparison. Okay, so now from this SAM file, so what I did next is, if you remember again the file formats, there is a format called GFF, and if you look at this GFF file now, this is what it is going to look like. And now remember what Dr. Paudel said that day, what GFF does. GFF is generally it has the general features file or the general features format. So it generally has different features within that gene. And on the second day, when we were introducing the coronavirus, we said, okay, in the beginning, there is ORF1, then there is different genes, S gene, N gene, E gene, and those kind of things, right? So this annotation file has all those. So if you look at this, it is saying 5' UTR goes from sequence 1 to 265. Then there is a gene from 266.5 to 2155. That gene, you know, you will see a lot of different ID here. And if you look at here, name, we are going to see name equal to ORFMAB, right? So what I'm interested is now, let's say S protein, because we know that S protein is very good for the infectivity, so it can infect and that work with the human cells. So I want to know what is the location of the S gene. Okay, now if you look at here, it has this name equal to S, right? So now you know that from sequence 21563 to 25384, this is our S gene. So that's what we know. And if you remember earlier comment by Mr. Subedi, I think there was a A to G mutation in 23,000 something base pair, right? And that is exactly where it falls in that S gene. And that's what we're talking about. Okay, so now the reason I was showing this one is the SAMtools view command you can also use. So a SAMtools view command has a lot of different you know, things it can do. So if I look at my history, I have done a lot of things. One of the things I have done is, and I think I would like somebody from our team to write this code, is that let's do this code here. We can actually filter only reads on that sequence from this position 21563 to 25384, and I'm just getting them as another BAM file so that it will be easier for me to load that BAM file or look at that BAM file. Okay, so that's why I made, and I just so sometimes what you can do is now you can just look at a single gene, what you are interested in, and make different things from that gene. Okay, so if we look at now that file that we created, the difference, oops, this is saying it doesn't have that file, so maybe something I'll see in a while. Okay, so okay, I think it has that one anyway. So we can see that, you know, where it mapped. It looks like it is for everything. It is not for just for the S gene. Oh, okay. So I think probably the reason is here because I did not convert it to the SAM. So this is that our SAM file for that gene. Okay. So what I did next is, now we know that there are things in that SAM file. We can also have a in the SAM file we can have a different quality, the sequence of the FASTA file and everything. So what I did is I took that SAM file and wrote all the sequences as a FASTA file. The way I did that is a very simple, that is not a standard bioinformatics command, but that is a generally Unix command, let's say, and that is I used awk command here. So what I did is, so again, this is not really you know, something we do in the beginning as a bioinformatician, but awk, you know, some basic bash commands and awk are going to be very useful to you in the future. Okay, so from that one, I'm not going to even show you that because it will, I think it will get confuse you. If somebody is interested, I can show you later. And then from that one, I have the reference genome. I have the genome from all of them, but just for the S gene. So the next one, and we are talking about the multiple sequence alignment. So the next one we did is use the program called Muscle, and use that program to make a multiple sequence alignment. And how we do that is Muscle dash in equal to this sgen.fa dash out it sgen dot. You can do, you know, MFA. I'm writing .2.mfa so that, because if it doesn't come up there, I can show you the earlier file and you can just enter it. So now what it is going to do is it's going to align this one. And so the difference between this alignment versus the other alignment is, the other alignment is alignment of short reads 150, 200, 300 base pairs to a genome of 29,000 base pairs in our case, but sometimes it can be a human genome, 3 billion genome, or the rice genome, right? In this example, the multiple sequence alignment is, it has tentatively same size of the gene, so and it just tries to align the best match with the other one. So it is something like, I think when we were kids, we used to play some games with our names, right? If my name is Ananta and somebody's name is Ananda, what's the difference? And we say, oh, okay, everything matches, and instead of T there is D. Okay, so it will take some time, so I'm going to kill this one, and I'm going to show how that will look like. So now if you look at that file, now that becomes a multi-FASTA, and that output format was multi-FASTA, and now it appends a lot of this NNNN in the beginning, because probably in some sequence there was other things, or maybe it started with the NN, because generally if it was the RNA was converted to the DNA, maybe we appended A there, and because of, and they truncated later and just said it's NNN. So if you look at this file, this looks all gibberish to you, right? It doesn't really make sense to you. What will make sense is, I also, when we are doing this this command, you can write a different format, and you can say HTML. So if you do HTML format, that is going to show in the HTML format, or you can also show, say, a clustral format or some other format that looks better for you. So I also did HTML so that it can show in the HTML format. So I already did that HTML, as you can guess, and I already loaded that HTML file here. Okay, so now this is what it looks like. If you look at this sequence, if you look at this sequence, it looks everything exactly similar, and all those different genomes we downloaded from there. And if you look at this one, it is almost exactly similar, but seems like there is a difference in this one. There it is calling NNN. Seems like there is no ATG there. So seems like there was that one difference. If you start looking at all these ones, then you are going to see, okay, it has all the C, but for one genome, it has that T over there. Everything was C, one was T. And all those kind of different things you can see here. So for example, there is another example. Seems like it was C for most of the genome, but it has T for 3 of the genome in this position. So this is how generally you visualize the multiple sequence alignment. Okay, so after we do the multiple sequence alignment, and it's not in a sequential order, but sometimes what we are interested in is, okay, yeah, I see here is one. So there are 2 ways we can use this information, right? One is like I did earlier with the VCF. Okay, we have the SNPs in this area, and this area. Okay, and also we have SNPs in this and this, and we can look at the function. And okay, so this will change the spike protein, so this becomes more effective, and those kind of things. The other one we can do is on the second day, I and also Mr. Subedi showed you the tree, and we showed, okay, seems like this is evolving this way. And in the evolution, the way we thought we did is we actually showed something that is already available, next strain, but we can actually make that tree by ourselves by just doing, okay, let's make a let's find out the evolutionary relationship between all these genes. And to do that, again, you can use the same uh, you can also use the muscle. And so you can see there is a command called muscle. It's still in the muscle. You can do a make tree command, and your input file is going to be the already aligned file, that MFA file you had earlier, and you can output the file as file format, or different formats you can have. And then we want to cluster with the neighbor joining algorithm. And again, there are multiple algorithms to do the clustering. I'm using the neighbor joining, just it is quicker than the UPGMA. Okay, so when I run this command, then I have this covid.phy file. Again, I'm not showing this one because it takes time, because it is a bigger one. That is the reason I'm not showing it here. But I'm showing those commands from my history to say that I actually did it. So if you look at this one, it looks like, okay, so how do you make information from this one? So it has different names, and this has these different numbers and those kind of things. And this is what we call a file format. And we have so many different programs to actually visualize that file format. We can use some software like R or Python. We can plot using different programs. All good thing is we all now have the access to the internet, and we can actually use the browser, our browser, and just simply copy that file. And if you look at the information here, this is what I copied exactly from that earlier one. This is not any different from what I copied earlier. So I just copy here, and then I am doing view tree, and I'm just showing, okay, how I want to show it and do it. Now you see this tree. Okay, so it seems like this is the case. We had one rooted here, then you see different changes, and it is showing up in the different level of going. So in this relationship is not based on the all 29,000 genome, but this relationship is based on just the S gene that we extracted earlier from mapping. Okay, so this is how we came up to the mapping. So now I'll stop, and and I'm pretty sure Dr. Paudel or Dr. Parajuli or Mr. Subedi, they are giving you all the course I'm showing, and you are probably working on that. So I'll give you some chance to work on your own files and also ask me questions. Is there anything you want to add, Dr. Paudel, Mr. Subedi, Dr. Parajuli?
I'll just have the code on the chat box.
Okay.
So we have country's name. So maybe we can just quickly look at it.
Yeah, that's a great idea. So
yeah.
Maybe we can, let's do this way. And I think I just did that earlier with the grep command, or I can do again here.
Okay.
Yep. Okay. Okay. So Nepal is 410301. Seems like this was the Nepal. 451958. Where is that one? Why don't I see that? Did we have more files than what we had
No,
originally?
it's Pakistan.
Oh, it's Pakistan. Okay. Okay. Oh, okay. Yeah, that's right. So this is Pakistan. Seems like Nepal's closest one was Pakistan. Probably I'm not sure when it was sequenced, but there is that one, and you can see the 434692. Thailand here. Yep. And then 450332 India. So I think that kind of makes sense. So if you look at this one over here, seems like a lot evolved here. 821
Portugal.
Portugal. Okay, okay. Or it might be one of those like, yeah, it probably it went to Europe and it's totally different. 418036 is USA. So maybe, yeah. So
Yeah,
I
pretty
think
interesting. The last one is Japan. The last, the 416569. Even though we are just looking at as gene,
yeah, yeah,
we're still seeing the geographical difference. Pretty interesting.
exactly, exactly. Yep. And S gene is not the very mutating gene. I think we are seeing more mutation in the E gene or N gene, right? Or ORF. So, yeah. Okay, I'll start looking at the questions. If there are any questions, we can. So I'll go from very beginning. Yeah, okay. Maybe some of the questions might have been already answered. So I'll just go through. Seems like the GTPL that is answered. Genome sequences from Subedi from Bangladesh by Child is a Wealth Research Foundation. I see that message to me privately, so thank you. So I'm not sure if that is from the CSRF, because recently I'm seeing a lot of the, or maybe they are doing all the sequencing. Recently, that is something that, you know, in the Nepal, we only see one sequence that was from Jan, but if you look at in NCBI, if you search India, Pakistan, Bangladesh, you will see hundreds of sequences. So that will show us where we are in the genomics research. Is that Python code available in repository in shareable form? If you are interested, we can make it available, I guess. Is that right, Mr. Subedi?
Yeah, sure. Yeah.
Is n equal to CGT? Or yeah, I see a lot of things are already answered. So since I don't have to go through all of those ones. Not sure if this is answered, but can we edit the sequence? So I'm not sure what you mean editing the sequence in the genome, in the live. That's what Dr. Parajuli is doing now, right? Editing the sequence, genome editing, all this CRISPR-Cas9. But if you mean on the text file, yes, we can do. We have not gone through that commands yet. You know, there is very simple command called vi or nano. You know, you can those text editors, but as a beginner in Linux, I generally suggest working in the Notepad++ or some other program and then editing there and copying that file over to the MobileStorm. What is the command for looking file after it's set tree created? Yep. So for tree, we have different programs. We can actually use the control from the command line. And I generally use some program from R. I have my own custom codes and use the R and the app library in R. But most of the time, if it's not a big deal, one or two times, I generally use what I did now. We go to some online, just ask file directory, and then just paste the file in the format, NAVIC format, file format, and we just take that screenshot here. What is the difference of when we use our computer storage and cloud computing in terms of using mobile extern? If you use your computer, you don't have to use mobile extern. However, most of the commands we see here are probably not available in the Windows environment. If you have a Windows environment, sometimes with the Windows environment, if you have the Conda environment, it will work with the downloading the Conda and then have all these commands. Some of the programs, but all the programs may not be in Conda. And so Windows is not really great for doing bioinformatics research. And so if you have a Linux computer, and for the sequence load or the programming capacity we are doing in this demo, this hands-on, it is enough. Even your laptop is enough. But think about when you are doing your real research in some big genomes, we should be using some high HPC, high performance cluster, or what high performance computing clusters, different clusters. And like a lot of organizations, I believe my organization, Mr. Subedi, Dr. Parajuli, Dr. Paudel, all of us have our own, not our own, our institute has their own HPC. We have petabytes of data storage. We have more than 100 gig network speed. We have probably, I don't know, 1,000 different clusters with more than 1 terabyte memory on each of them. So, if that is really expensive, but if we are not sure, we always can rent something from Amazon Cloud, Amazon or Google or Microsoft, like we are doing today. And the good thing about this one is we thought, actually this is something interesting. We thought in the beginning we'll only have 10 or 15 people. So we thought, okay, that is enough. And we made a smaller cluster, smaller memory, smaller size. But after we saw your response, we actually increased it because we thought, okay, we need more memory. And that is why I think Dr. Paul said today in the beginning, probably we don't have to worry about that today because we increase that power, because so that we can accommodate you all. So that is why the working on the cloud is very flexible. What is the difference between ET toolkit tree viewer and CBI tree viewer? Probably no difference. Probably ET toolkit just came when I searched the Google. That was probably the first term I saw. Okay, so I think that those are all the questions. So have you been trying? Let us know, have you been trying and where you are, if you were able to copy some of those codes and run those ones. Let us know here, or you can, I think you can unmute yourself and talk about that. Do I need to copy my FASTQ file to the working directory to use it for easiness? Generally, yes. We have to have it in the we need to have it in your working folder, but there are always ways. We generally work to make it cleaner. We make it all. We have one working directory called COVID-19, and then we have raw FASTQ files in one folder. We'll have another alignment folder where we'll have all these SAM and BAM file and those kind of things. Generally, you have to have some folder structure. Can we show BP count on multiple sequence alignment? Yes. So we can use that WC command, the word count command. Mr. Subedi showed you yesterday. Is tree viewer equal to dendrogram? Yes. So I think in short it is yes. People judge, we can go and say no, this tree and dendrogram mean different things. For the UPGMI, we don't call it dendrogram or those different kind of things. But for our purpose, the answer is yes. So it's about 1 hour. Let's take a little bit break, and by break I mean I think today is the last day. So I would like to invite Dr. Himal Notel to update us a little bit about what the AFU Biotech Center is doing, and what do you think of this program, and how can we collaborate more?
Thank you very much, Dr. Ananta. Sorry for disturbance. Good morning, good afternoon, and good evening, according to the respective time zones, as we have participants and experts from all over the globe. I really want to thank the experts, Ananta, Acharya, Sishir, Subedi, Dev, Paudel, Dr. Bartola, and Saroj Parajuli, and your dedication for these 5 days. This was since last year we started some sort of outsourcing for bioinformatics in the AFU Center for Biotechnology, and later on we came into idea of this kind of seminar come workshop, and I think it's really worthwhile for us, and we see a lot of encouraging participants right from 206 in the beginning, and then still we have 115 participants actively learning at least some insights of bioinformatics, and later on we want to really conduct the compact training program. This is for free, but later on we want to make it with some paid version of trainings, hands-on training. We'll publish the notice after discussing with the expert panels. And regarding the Center for Biotechnology, it was started since the beginning of this university in 2010, but till 2014. It was not active enough, and in 2015, myself and one more faculty member, Ananta Sharma, who is also in the Zoom meeting from Canada, who is doing PhD, we started at least some sort of lab establishment. We didn't have so many equipments in the beginning, and during that time, our director, Professor Dr. Bhuminanda Devkota, he supported a lot, and later on we still have IP Acharya as a director, and we worked together and we brought lab into at least one level. And at the moment we have a molecular lab for plant and animal side. Likewise, we have mammalian cell culture, and we are starting tissue culture facility here at AFU. And recently, we are really fortunate that we received the competitive grant from University Grant Commission, almost $1 million for project for equipmentation of the center. And in this, this is called Integrated Research Laboratory, IRL. And within this IRL, we have 5 distinct units that are envisaged, and that will be established by this year. And we will have very common shared facility for all of the laboratory units. And regarding the laboratory units, we will have molecular biology both for plant and animal that we will upgrade to what we have. And we will have a microbiology unit which will contain the virology as well as bacteriology lab. And we have primitive mammalian cell culture lab. It will be upgraded to the dedicated cell culture lab and embryo culture lab. And recently we started the tissue culture lab, and we will upgrade it, and then it will be fully functional. And one more novel approach we will introduce into our center is the analytical lab, where we will have UPLC and mass spec, and we will do a lot of residual analysis, pesticide analysis, and identification of different active compounds from different plants throughout the country. And will study the perspective of medicinal use of those plants. And academically, we are running masters in MBSc, Masters in Veterinary Science Biotechnology, and MSc Biotechnology. And in near future, we will start probably by this coming academic year, we will start the Masters in Bioinformatics. That will be really in collaboration with NAPA, and then that will be one of the innovative approaches of the center. And likewise, once we install UPLC and mass spec, we will start masters in analytical chemistry as well. So that our center will be really active enough in these areas. At the moment, we are not planning to conduct the undergrad, only the graduate schools will conduct. And our lab will really focus on three things. One is the academic programs, of course, this is the heart of the university. And then we will focus on research, and we'll focus on continuing education. So this is one of the events that continuing education, and there will be other training programs for wet lab as well. And according to the expertise, we are working on different areas like genetic analysis of indigenous as well as crossbred animals, that is led by Dr. Acharya, and myself, I'm more engaged on poultry pathogens, and Professor Madhav Pandey, he's engaged in the plant molecular lab, and then working on wheat and rice, and as he mentioned before as well. So these are the areas we want to cover off, and I hope some of the experts present in this meeting will be well connected with us, and then in future we will have a lot of collaborative approaches to work in this area. Thank you very much. We will continue the session in a moment. Thank you, Ananta.
Thank you, Dr. Nigel. So there are some other questions here, so we want to go through that. One is on Mac again, how to connect with the Mac and those kind of things. We went through that yesterday. I'm not sure if you were there yesterday. So you need the .pem file, and Dr. Parajuli showed that how to do that yesterday. And I think in our tutorial, I think we already have that, or if not, we'll make sure that we'll add how to connect to the terminal. Also in our tutorial we'll be sending out today. The other one is how long this server will be live. I don't know the answer to that one. So we have some funding from AFU Biotech Center. So it will depend on when we'll run out, how much you will use, and when we'll run out from that cost. So it will probably depend on that. But I would hope that at least 2-3 few more days we can. It will be on. There is also a question on how to upload to the NCBI or NCBI. And if it is about the sequence SRA or the FASTA files, you created something new data, something like this, the coronavirus data you want to upload for users, then this is kind of a complex process. You have to have a bioproject, biosample. It takes a little bit of the effort. It's not as easy as transferring. However, you mean the way I just uploaded to the E3 viewer, then this is simple. You can just right click that file, you download to your local computer, and then download, right click and open as a notepad or notepad plus plus, and then you can just copy that text, or you can just download that file and upload to that. I think I just saw the NCBI user, there's an option called upload file, so you can just use that upload file and upload that tree file. Okay, so this is kind of the overview on all the genomics. I know this is kind of a rush through. I know you couldn't, probably not all of you, could do all the steps, but this is the general introduction. And after you are done with, after we are done and when we send out the code, try it. I'm pretty sure you can do it, because today we saw how many 120 or so accounts, and some of them had the past few files or SAM files. So that means, yesterday by end of the session, we had probably 40 or something. So that means you were doing it, you did it great. So thank you very much. Next, we get a lot of questions, because this is also about the biotech and not just the bioinformatics. So we have a little bit of time. So it's kind of both the combination of the biotech, how we apply some of the bioinformatics and the biotech. So and it seems like we saw some interest about the SnapGene yesterday. So I would ask Dr. Parajuli to show us, if let's say if we have to start our PCR lab for the coronavirus, how do we do that? How do we make different primers and start making the PCR, making the primers for the virus?
Okay.
So, yep.
Thank you, Dr. Acharya.
We'll go through that one.
Okay. Welcome back to all of you on the final day of our workshop. So we are moving a little bit away from bioinformatics. So we are going to core molecular biology. So what I'm going to show you today is like how to get the sequence and design the primer and do PCR. But one thing is, like in this SnapGene viewer, I cannot show you because this is like, it has to be a paid version. So if we have paid version, we can design primer, we can simulate the gel, and we can see how it can be seen in the gel by using different enzyme or by doing PCR. But here I can design the primer in the website, and I can bring that primer to SnapGene, and we can do some like how it comes in the PCR or what will be the size. We can show a lot of things. So if you have not downloaded the SnapGene, and if you are interested, first thing is you can download SnapGene. Let me share the screen.
And while Dr. Parajuli is sharing the screen, we just sent out a survey to all of you who registered for the program, and also I think we included some of you who emailed us about the different codes and all the probably not registered. So please fill that survey. It will be great. If you can fill that survey after, by the end of this course, or now we don't want you to distracted, then maybe 10, 15, 20 minutes after the course. So that will be really great. And if you have not got the survey link, please write your email address in the chat box. We'll send you that survey. It will be really helpful. Or maybe I think, is there a way that Dr. Paudel, can we just link that survey link here, and they can just click on the link and provide survey?
Yeah, I'll just do that.
Yep.
Can you see my screen?
Yes.
Everything, right? The whole screen.
Yeah.
So first thing is, if you have not downloaded SnapGene, we can go to SnapGene to download it. So we have to type SnapGene viewer. And if EFE Biotech unit is looking for some software, then I think this will be one of the best software, so that you can do so many things. And you can do a lot of things in the free version also, but if you pay, then there are so many different things you can do. You can clone, you can simulate jail, you can do a lot of things. You can do cloning, different type of clonings too. So when you go to the Snapmachine Viewer website, so go on the bottom, and according to your operating system, you can download it, like Windows, Mac OS, or Ubuntu. And if you have any trouble downloading, you can write in the chat box. Okay, I'll wait a few minutes till you download and install it. So in that time, I'll go to the NCBI website to get some primers so that we can design. So let me open a new NCBI browser. So in plant, Rubisco is very famous enzyme. So I'm going to design the primer for Rubisco. You just type Rubisco in NCBI. There are 5,000, more than 5,000 genes for Rubisco. So if we want, we can select based on a specific organism. If not, we can just click any of those enzymes. Maybe I'll go for Arabidopsis thaliana. Let me know if I have to slow down or stop for you.
Slow down, zoom a little bit.
Let me open NCBI again.
Zoom it. Zoom out.
Okay. So I'll paste the link for NCBI also. Let me do that. So here is a link for NCBI. And after you go to NCBI, first thing you need to do is type the gene of your interest. So for now, we are interested in Rubisco. So there are more than 5,000 genes. You just click here. So you don't have to get the same gene. You can get any sequence because like we are designing the primer. So any of the sequence is going to work. So I'll get this one. And if you are not able to type Rubisco, then one direct method will be just, I'll copy this one in the chat box so that you can go to NCBI, type this one, and you'll get the sequence. So there are a lot of information right here. You can see so many different things, but we are interested in FASTA sequence. So I'll scroll down and there are gene bank sequence, FASTA sequence, but we are interested in FASTA. So I'll just click FASTA. And then I'm going to copy the sequence. Now I'm going to paste the sequence in the Snapmachine Viewer. Let me know if I have to wait. So to paste the sequence in the SnapGene viewer, you have to create a new file. So I'll go to File, then create a new file. So we are making DNA file. We are not, like, if we have to copy the protein sequence, then we can make protein file also. But we are making DNA sequence, so we'll go to DNA file, and we'll paste the sequence right here. And you can give a name. So 80 for Arabidopsis thaliana, and I write Robisco. And one thing you can do is, like, based on the sequence, you will know like it is positive strand or negative strand. And if you have to change the strand, you can go to reverse complement. It will just read from the back. I'll click OK. Now I can see the file, and let's see how long is the sequence first. So it is 3,579. Let me zoom in. So we can see right here, it is 3,579 sequence bases are there. Let's see the whole thing. So on the top, it says it has 40% GC content. So you can see the GC content for the whole file, or you can just see for the few sequences that you are selecting to. So if I select this much, so if you can see right here, so the GC content will go on changing as I go on selecting the sequence. So if you want to design the primer, and you can see how much is the GC content too. And first thing you can do is see the map, you can see everything, you can see the enzymes. And suppose if you are doing restriction digestion with a single enzyme, then suppose you are working on BSA1. So if you click BSA1, then you will see like it is cutting at only one location, that is 532 base pair, that is the location where it cuts. And if you do gel electrophoresis after cutting with this enzyme, then the whole segment is going to be divided into two sequences, because the restriction enzyme is going to cut right here. And I think like most of the sequence here are single cutter. So we can change the enzyme, and we can go for double cutter also. So let's do whole enzyme, or just go to commercial NEB. Okay. Suppose we are interested in PLE1 enzyme. Okay. So if you are working on this gene and you want to see what will happen after you do the restriction digestion with this enzyme, then you can see it has cut into two different locations. One is at 464 right here, and another location is 2828. So if you use PLE1 for restriction digestion, then you are going to get 3 fragments in the gel electrophoresis. And suppose if you want to clone the one that is cut by PLE1, then you should be looking for the size that is the biggest. So that is from right here to PLE1 right here. So it will be around 2400 base pair. So if you have to elute the DNA fragment from the gel, then you have to look around this size in the marker. So when you run the gel, you run with the marker, and you have to look for this size, then cut the gel out and elute the DNA fragment and go for further cloning. But now we are interested in designing the primer, and this is not free version, like this is not paid version, so I cannot design here. I have to go back to another software online. So can you see the website, right? Dr. Acharya? Anyone? We can see website, right?
Yeah, we can see.
Yeah.
Okay, I'm going to Primer 3, very famous software for designing the primer.
Can you zoom a little bit?
Okay. So let me go back then. So we are getting the sequence from here. And one thing is, suppose we are plotting with this enzyme right here, PLE1. So what will happen? Maybe we have to design the primer only inside this fragment. So let's take the sequence only from this fragment. We'll go to sequence, and PLE1 is right here. So we can just get in between the PLE1. Let me go back to map. It will be easier. Okay. So I think that's selected. So I'll copy that region. And I'll go back to primer 3. And in primer 3, on the top, you can see a box where you can paste your sequence of interest. I'll just paste it. And on the bottom, there are so many different parameters that you can set based on your requirement. Like you can set a target, you can exclude a region where you don't want to design a primer, or you can set a size, like how much will be your size. And suppose if you are cloning, then maybe you are not interested in size. Maybe you want to just do from the two end. But if you are doing PCR or maybe like qPCR, then maybe the size you need will be like 80 base pair to 100 base pair or 120 base pair, not more than that one. So based on your interest, you can set the primer size. Sorry, size of the product, not the primer. Primer will be around 20 to 23 all the time. So I'm going to set 100 to 200 now. So we'll see. And also you can set the temperature, like how much you want. So it is default, so I'll just leave it like that. And same thing with the size of the primer. You can change according to your need. Like if you want a smaller one, then you can like change it to like 15, but that is not recommended for higher genome organism because the primer could be repeated. So I'll just leave everything as default except the product size. Then I'll just go and pick the primer. So, okay. So it will show me the one pair of primer on the top and like where it started and what is the length, melting temperature, like GC content, oh, exactly 50%, okay. And 3' sequence. Sorry. And the sequence of the primer. And if you go on the bottom, then it like sometimes this primer may not work for you. And sometimes people design more than one pair. And you can find other pair on the bottom. So based on the requirement, we can select the primer from here. And if one pair doesn't work, maybe the other pair is going to work. So when we order the primer, we order more than one pair because like if one pair doesn't work by any chance, then at least we have a one backup. So for now I'll just use the first pair of primer that we have. So zoom in. So this is the forward primer from the 5' in. So I'll copy this sequence and go back to SnapGen viewer. And there is an option on the top called primer. I go to primer and do add primer. And you can give the name of the primer. It is Rubisco forward 1. So we can have multiple forwards. So we give 1, 2, 3 and add primer to the template. Okay. So we got Rubisco forward 1 and we'll go back to the web page to get the second primer. And go back to SnapGen viewer again. Same thing, I repeat the same thing, add primer. So it will automatically detect that is from the lower strand. So I'll give the name of the primer, Rubisco reverse 1, and add primer to the template. So this is the reason where we are designing the primer. So let's check what is the size. 154. So if you are interested in this fragment of this gene and we have designed this primer, then after doing PCR and running the gel, we have to look for 250 base pair fragment. And we can, like, if we just want to know the variation of the size, like people will be working on marker, then there could be some insertion and deletion. Based on that one, you can identify the size too. Because if there is some insertion or deletion, then the size will go lower or higher. So based on that one, you can differentiate the genotype also. And if there is some gene of interest and you found there are some SSR in the genes, then in different SSN, you can run this primer. And to run the primer, first we have to design the primer from the consensus region, because the primer has to match in all the SSNs, otherwise this is not going to work. And based on that one, you can run the gel and you can finally get your fragment and determine, okay, this belongs to this type of SSNs, or this is not from this SSN. You can decide on those two. If there is a admixture or any other thing, you can identify those two. And one last thing is, like, if you are interested in sequencing, suppose you know there are some SNPs right here in this position, then you can design the primer based on the SNP, like forward and reverse from the SNP, and do PCR, run the gel, and get the fragment. Then you can send only this fragment for sequencing. Like when we did the genome, like we got 150 to 200 base pair long fragment, and we go on aligning to the reference genome. Similar wise, if we send this sequence for sequencing, then you can get 150 base pair, then you can see what is the variation in the sequence. That can also be done. And one more thing is, okay. And also in SnapGene Viewer, since you have the, like, you can see different enzymes right here, then you can see if you use some specific enzyme, then what is going to happen in your fragment, if we are interested. If not, then mainly like we look the enzyme for the cloning. Otherwise, we just run the PCR, run the gel, and we'll know the fragment size. So this is the maximum magnification I can do here. And I have already told you, like, if this is a paid version, then we can do so many things. You can just run the gel right here. So yeah. There is a option called simulate agarose gel, and based on your sequence, you can run the gel based on the, like, either primer or maybe the enzyme. You can run the gel here. But unfortunately, we cannot do it here. But we can do cloning here. PCR, not available. It says like only on the full version. So let me know if I have to repeat anything from here, or if you have any questions. Can we use SSR-based amplified fragment product sequence for variant calling or phylogenetic tree?
I think I'll take that one. So for variant call, SSR is mostly designed after you already know the variants. So you would already know the variants. You already know what are those repeats, and then based on that you design the primer, and then you can score your SSRs, and then you can use it to make a tree, but not for the variant call itself. I think
One more thing I can show you is like we can get multiple sequence and align in the website if that is of interest. We can do multiple alignment in the, like there are so many things, like if you are not working on the genome level and you are working in the level of the gene or like some mRNA transcript, anything, then you can do most of the thing in the website. You don't need to like fancy softwares or any other thing. We can do most of the things online.
Yep, and I also encourage you to explore something called Galaxy. So a lot of the things we have in this bioinformatics session, you can use the Galaxy to do a lot of the things we talked about in just point and click. But it is, you know, it will be one by one. You cannot batch it a lot. So the way this bioinformatics and most of the programming works is, after you are used to this programming and working on the command line, you almost hate the point and click. Sometimes my days goes without, you know, touching a mouse. So, you know, that's how you know, we use. So I think we have half an hour more, so I think we can start wrapping up this one. So I think to wrap up, I think I'll invite all the speakers to give their experience on last 5 days. What was our expectation and what was our experience and how we can collaborate more. And also, I would like to invite in the end, I would like to invite Dr. Kodaria again for this opportunity and how we can collaborate more. And then, if from the audience, if somebody wants to give the feedback, we are seeing a lot of feedback in the chat room. We are also already seeing some of the survey feedbacks, but if you want to give it verbally, you are welcome to unmute yourself and talk. Let's end the last half an hour. Let's just celebrate our 5 days session in last half an hour. I'll start with somebody from our experts panel. So Dr. Parajuli or Dr. Paudel or Mr. Subedi, please go ahead, give your opinion on this last 4-5 days, and then we'll go from there.
Okay, I can start. Okay. So in the beginning, when we planned to do this type of workshop, and we thought maybe we'll have maximum of 100 people, right? Then after a few days, we saw like the number went on increasing, and we were more excited because like it was far better than what we expected. And now it has given us a lot of energy that maybe we have to do these sessions frequently in the future, maybe like in every 3-4 months or 6 months, or based on the requirements. So if you are interested in any of the things that we have been doing, and if you want to learn more, so don't feel shy to reach out. So you can reach out to Dr. Luitel or Dr. Acharya or Dr. Paudel, anyone, just reach out to biotech unit there, or you can reach out to us. Then we can deliver the lecture based on your requirement. And I'll repeat one more thing, like what we have done already in the Linux commands, same thing we can do in website, we can do online. So if you are not working in big genome and if you just want to know a lot of things, so go on website, search for so many things, then you will get a lot of information from there. You can download the sequence, you can align the sequence, you can design the primer, you can do so many different things. And like if you are stuck somewhere, you can come back to us and you can send email or send message, and we can help you in any way that we can from here. So that is our main goal, like to help you all in like what you are doing, and encourage you to do more bioinformatics and learn so many different things. So we are also coming back from the same background, and like when I was studying masters there, and like we used to talk about markers, and like we had theory knowledge, but we did not know like what actually it was. So if somebody had given some opportunity to learn or to know some things better, then maybe we would learn so many things at that stage, then we could have done much better now. So it is your turn. So take the opportunity and learn as much as you can. I think that's all from my side.
Thank you. Dr. Paudel?
Thank you, Dr. I want to reiterate everything Dr. Parajuli and Dr. Acharya have mentioned about how we realize and how we feel that we're really successful because, yeah, and again, that we got a really overwhelming response for this thing, and we are really glad more than 100 people are still sticking out until the final day. It has been tedious. I know there were a lot of new things for most of you, and it might have become overwhelming. We have tried our best to keep it as simple as possible, but since we have been working this for quite some time, we might have used a lot of jargons, and even though we didn't want it to happen like that way, we might have made it a little bit more difficult. But we have also shared all our codes that we did during the session, so you can go to the link that I provided and just follow all the steps that are there. So you should at least be able to at least download data from NCBI, be able to uh, create a fast QC and like check the quality. You will also be able to do alignment, and then uh, from today you will also be able to do the phylogenetic. I was looking at some of the that you already worked, and I could see that some of you have already made a phylogenetic tree also. So I'm really glad that at least there are a lot of people who were able to copy us from the beginning to an end, and I think that that is a huge success. I want to thank all of you for your continued cooperation, and please do fill out the survey form that we have sent you. This will be really important. So if you want us to continue doing this type of workshop in the future, please make sure that you write and show your interest there. And any other feedback to improve this seminar would be really appreciated. And finally, I also want to Dr. Acharya and Dr. Lutel, who have been helping tremendously in making sure that this program runs smoothly. And I hope that we will be able to collaborate in the future. Thank you.
Yeah. Hello. Yeah, I would also like to start with thanking AFU, Dr. Kadaria, Dr. Luitel, Dr. Acharya, Dr. Paudel, Dr. Parajuli, everyone in the committee and everyone. This was a really great experience, learning experience for me as well. And I hope it was also a good learning experience for all of the participants. And the main thing we were really excited about is the number of participants. Initially, we thought that when we saw there were about 100 participants, we thought it was already successful, and now we're running around 300, and still we have 150 in second day, third day. So that was really something to note, and to in future that there is interest among participants. So we are really excited to keep continuing this sort of activities in near future. And so especially for the newcomers, one message I would like to convey is that it might seem really complex, like a lot of things going on, programming, and especially if you are coming from bio background. So, and as Dr. Parajuli mentioned, that we all come from same background, and main thing here, what worked out for me is to have patience and be very take things step by step, and you will be really surprised at how quickly you learn. Not to get overwhelmed with so many things and technologies. After all, it's just a biology, and you're just trying to get more information out of the tools that you're using. So just have patience, and you can contact us and other professional in the field. You can watch a lot of YouTube videos, a lot of resources are available there. So just, yeah, keep working hard, and best of luck for everyone. Thank you.
Before I would ask Dr. Kadaria next, and I also want to hear something from maybe some of the past students from last semester or earlier, that we kind of went through similar work on the hands-on experiment, and what is the difference that time versus now, if you can share some of the experience, that will also help us. So if somebody is also attending this one who attended my previous hands-on courses, please provide the feedback. So while you are thinking who wants to do that, Dr. Kadaria, please.
Thank you, Ananta. It's a great success for us, both NAPA and EAQ Bioinformatics Center. We have completed our this hands-on workshop, 5 days hands-on workshop in a totally new area for us, for its Nepalese community as well. Participants, our AIQ and other participants from different institutions. It's genomics, it's totally new for us, though we as a faculty, we know something, but in depth knowledge, we should borrow from experts, or we should outsourcing these experts. So that's why you people, you guys, you did a lot for us. So first of all, I would like to thank you all, expert panel, and then NAPA, and then our head authority, and then without participants, it wouldn't be a success. So also I would like to equally, participants are equally important and equally valuable for us. So in coming days, what we are thinking, we will continue. It's our commitment. We will continue such type of training. Even we are thinking in type PCR wet lab training, like PCR, normal PCR, qPCR, cell culture, or different protein level wet lab lab work level training. We are thinking, and we will organize such type of training. So participants who you are listening us, so please keep on your eyes open, and then we will keep in touch with us. If you ask us, if you create demand for us to take such type of training, we can organize in coming days. And in regarding this biotechnology or bioinformatics, so discussing with this expert panel or NAPA or our authority, we are thinking to start some bioinformatics courses. Even if possible, we will start separate postgraduate level course, bioinformatics course in bioinformatics in PG level. So for this, we have to prepare some policy process. So okay, so we are also thinking some genome level work and research work, like some our this genome work in Nepal is really virgin area, and then sequencing is very less sequencing are done in Nepal. So for like indigenous cattle, indigenous buffalo, sheep, goat, and like other poultry level, and then crop, even many our valuable crop species are not sequenced yet, and then are not analyzed by informatical level, and then this DNA, sorry, genomic level. So we will continue such type of work together. So for such type of work, some innovative students, some hard work students, it's just a kick-off. So in coming days, you all students, if your eagerness, if you show such type of eagerness, which you are showing to learn more and more about this on this subject, so we will facilitate you, and then we'll connect you other more experts, and then I already told you, like we will continue such type of training in wet lab, and then such this computer based training. So it's okay. Again, I would like to thank you all. It's a great success. We are very happy, and then keep in touch, all of you. We will continue it in coming days, and then my team, Dr. Himal, and then I especially thank to Dr. Ananta. It's a very basic discussion, both as a brotherhood relation I have with him, and then just started a single discussion, and then it materialized a big deal. And then coming days, it will go in next level, next height, and then it's good. Thank you all. I'm trying to connect with our vice chancellor to put some remarks, but she is not connecting yet. Okay, thank you.
I am here, Dr. Acharya. Dr. Acharya.
Dr. Thapalia, we can hear you, but seems like audio is a little bit problematic. I'm Ananta, by the way.
Hello, Dr. Acharya. I am connected. Okay, there is no electricity now, and I have connected with data of my data region to this.
Yeah. Now it is working better. Now it is working better, ma'am. You can speak.
Okay. Okay. The are you hearing me?
Yes, Dr. Thapalia. Yes, ma'am.
I will keep my video off because the internet is a little poor here. So thank you very much. Very warm good morning to everyone. Though I could not attend all the program, I was taking information from Dr. Acharya and Dr. Himal, and it was very successful and very interesting. I came to know from them. The 5-day hands-on workshop on applied bioinformatics in agriculture and medicine has been completed successfully. This program was jointly organized by Center for Biotechnology, AFU, and Association of Nepalese Agricultural Professionals in America. It is now apparent that there is a huge interest in this subject. The invited speakers shed light on many core issues of this field. I hope all participants have made most of this golden opportunity to learn from experienced members of the community. I thank all the invited speakers. The things we have learned in this workshop will help participants spread across the world immensely. As Vice Chancellor of Agriculture and Forestry University, I know that our alumni will continue to achieve great things and prove their merit all over the world. We can further enhance the strong network of agricultural professionals who love their motherland and Rampur. And I hope your network grows bigger and stronger. We can try to connect with each other and every Nepalese agricultural professionals and create a huge network and that can work together to help each other professionally as well as personally. NAPA has experts in vast number of fields with substantial research and professional experience. Technology has enabled us to share our expertise with people all over the world in new and exciting ways. I believe AFU also can benefit hugely from the support of all distinguished professionals in this network. Our university will try to make the most of the possibility in this regard. I hope we can further strengthen our relationship and cooperation for mutual prosperity. I would like to thank and congratulate all once again. Thank you very much for providing this opportunity for me also to learn many things about biotechnology and bioinformatics. Thank you very much.
Thank you very much, Dr. Sada Thapalia. It is really encouraging to hear from you even in the last day. And on the first day, we expected and unexpectedly we heard from you. That is really great. It really encourages us. So like you said, we'll get connected and we love Rampur and we love Nepal. So we will collaborate more. Thank you again. Thank you, ma'am.
Thank you very much.
Okay, I think with that, we are coming towards the end of the session. Anybody has final comments, Dr. Paudel?
Okay, I want to learn something from participant. I want to know.
So for any participants, if they want to speak, unmute yourself and speak. So that will be great. We know you are also providing feedback in survey. That will really help us. We are already seeing very good, very great feedback. So.
Hello, I am Kaisar Shehzad from Pakistan.
Hi, how are you?
Yeah, I have done PhD from Guangxi University, China. Actually, my supervisor, Professor Yang Qinglu, he recommended me to attend this seminar. And I am thankful to the NAPA team and the AFU Nepal for organizing such a tremendous and successful seminar. It's my hope that in future you will organize a seminar on RNA sequencing analysis and data visualization. Yeah, that's it from me. And thank you so much, organizers, again.
Thank you, Keshav. It's great to hear from you. And thank you for joining from Pakistan and China. And so that's great. Thanks. Okay, there's a question when the participants will get certificates. So we are preparing for that. So we'll based on some of the surveys you have sent, and based on some of the feedbacks and what we see, and I think we also captured some of the things here. We will also looking at different your participants in the Zoom meetings. So we expect at least 60-70% of the participation. So that kind of thing. So we are preparing. By any chance, if you don't get the certificate, let us know, and we'll see about that. Any more participants?
So regarding the certificate, we are analyzing the attendance as well, because we cannot issue that certificate for all 402 participants because they are not connected. So normally we discussed even earlier that at least 70% time is spent in this seminar will receive. We'll calculate right up to today, and then we'll issue the certificate. If somebody has 60%, he or she will not get it, that's for sure. And feedbacks will be really helpful for us for future improvement. Actually, this will be more encouraging because nobody and none of the programs can be perfect, but we want to try and make it better. And for that, your really critical suggestions are helpful for future improvement. Thank you very much for all the participants.
Okay, I think with that, I'll provide my final comments and then we can close the session. So I think I have said this time and again, time and a lot of all the instructors or co-organizers, AFU, everybody is saying that we are really excited. We thought it will be less than 100%, but it became, and we were just laughing, we're saying on the 5th day there will be maybe 5%, that's where we are saying, but we see in the 5th day more than 100%. That is really great. So first of all, I'd like to thank all of you participants and your interest. We have seen so many questions, and we all know that we rushed through this one. The reason for rushing through is just to introduce this to the topic. We did not expect you to learn everything in this 2-day session and be able to do everything, but we just showed some commands, and in the future you can use some of our tutorials to go through the commands and at least show you what can be done and what is the sequence of analyzing the data. And this is also just one part. Most of the bioinformatics research, we kind of follow the reference genome, basic FASTQ, mapping, and variant call. These 4-5 steps we do for almost every bioinformatics application. And what we do after that are different, whether we look at the phylogenetic tree, or we start on the in the cloning, or in the gene editing, or we are in the genomic selection. These and mapping, the QTL mapping or association mapping, and those different kind of things. So that all the different will be different. All the metagenomics on the field of the human health and other kind of things. So whatever you learn today, it will be applicable to anything that has either RNA or DNA. And again, thank you to all the participants. And I also would like to really thank you all my, all our team, like Dr. Kodaria said earlier, it was we kind of collaborated earlier to provide some course, and then okay, let's do something a little bit bigger and for the mass level. And then I was like, I might be too tired for like 3-4 days, so let's find some collaborators. And it's great that I had these great collaborators here. Sangeet, Sishir, and I'll use their first name because they are so dear and near to me. And so Sangeet, Sishir, Dev, Saroj, Radhika. So you all helped so much, and without you, this program would not have been here. Then I also like to thank our keynote addresses, BC Sada Thapalia, and also Dr. Meg Parajuli. They provided some collaboration ideas, and they really encouraged us. And I'm really encouraged by the speech by Dr. Thapalia today. Even she came today, just we on schedule. We just invited her, and she came, without early notice, and she gave us such an encouragement to us. So again, I would like to thank you, thank her. And then I would like to thank you, Dr. Himal, Lutil, and Dr. Kodaria. So without their involvement or even the initiation, it wouldn't have been here. So without your help, we wouldn't be here today. So and Dr. Kodaria, you both and Dr. Kodaria, you are doing a great job leading this effort. So thank you very much. And the final thanks to the whole AFU, Center for Biotechnology, and also NAPA for providing some support, like for example the Zoom support. Because we have like 300-400 participants, our basic license could not work. Also to the AFU Center for Biotechnology, providing some of the cost for all this computation and server. So it's the effort of all of us, different persons and different capacities, and it really became a lot better than we thought. Yes, there are some room for improvement. We rushed through few things. We kind of had evolution, genomics on animal, coronavirus, rice, and all those kind of things. But that was on purpose, so that we can have that wide audience, just to show them, okay, what we can do on different fields. But ultimately, if we do in the future, we'll probably focus on one subject or one area. So I think with that, and again, thanking all of you and the participants, and good luck to all the participants, and please contact us for any collaboration ideas. Dr. Kodaria and Dr. Lutil were saying already, we might have some wet lab in the AFU, but even without wet lab, you can use a lot of the public genome, public sequence, and do your research on bioinformatics. So you can actually probably, if we see some commitments from Dr. Kodaria and Dr. Thapalia, that we might even have a bioinformatics master's program. In that case, we might be able to do some of the thesis work without even generating the data by ourselves. Maybe we can use some public data. So that will be really great. So keep your interest on. It looks overwhelming, but again, like Dr. Parajuli said, we were on your shoes 5 years ago, 10 years ago, or 15 years ago, and we can all do that. So with that, thank you everyone. And let's celebrate the success of this webinar and the session and the whole session. Thanks. Thank you everyone.
Thank you everyone. Bye.